Hi, everybody. Welcome back to the Human Cell Atlas meeting on COVID-19. We're going to start our second session today, which is Beyond the Airway. Anna Greka. Anna is from the Broad Institute, and she's our next speaker on SARS-CoV-19.
Hello, everyone. It's a pleasure to be with you today. I'm working on sharing my screen. Can you all see my screen now?
That's beautiful, Anna.
Excellent. So again, it's a great pleasure. Thank you, Musa, for the lovely introduction, and to the organizers for inviting me to speak about the secret life of SARS-CoV-2 in the kidney and beyond. And I'm hoping I'll be able to explain what I mean by this in this short talk. Just starting from this article in the New York Times in April of 2020, really at the peak of this coronavirus pandemic, it was discussed how kidney disease and the need for dialysis was an overlooked and possibly fatal coronavirus crisis. And we will discuss how this was sort of a multifactorial event, both based on patients having severe respiratory failure, but also requiring additional support, as in the form of kidney dialysis, which is a system for supporting patients who are experiencing kidney failure. And so, I can attest to this during this time. While I spend probably 90% of my time doing work as a scientist, I am a fully credentialed physician, and I volunteered, and so was asked to serve during this time. And I can say that in Boston, at the Brigham and Women's Hospital where I work, we had to triple our acute dialysis service in the ICUs during that time in order to take care of the surge of patients we needed to take care of. So clearly there are stories around SARS-CoV-2 and the kidney that are worth discussing. This is from a study in the New England Journal that basically describes the significant amount of viral load that can be detected in the kidney, of course, second to the lung and pharynx as shown here, suggesting again that perhaps direct effects of the virus on the kidney should be considered, in addition, of course, to indirect reasons why the coronavirus COVID-19 might be affecting the kidney and causing kidney failure. Now, through the work of the kidney and lung HCA networks, and through the work by Shreya Subramanian, Catherine Vernon, co-supervised by Aviv Regev and myself, we were able to do some work to try to identify the expression of ACE2 in the kidney. This was part of the broader effort to look at multiple tissues. And this is no surprise. This was also work identified also by colleagues at other institutions to basically see that, as I show you here, the structure of the nephron, the functional unit of the kidney, the ACE2 expression is really quite abundant in the kidney, especially in the tubular compartment, which is shown here. Which may in part explain how direct effects of the viral entry, to the extent that virus makes it through the circulation and travels to the kidney in the most ill of patients, may in fact be able to enter the kidney through the expression of ACE2 and cause harm to the tubular epithelial cells. It is exactly the loss or death of these tubular epithelial cells that correlates with the presentation of acute kidney injury in our patients in general, as well as in the COVID-19 patients. And so this, to some extent, makes sense. One would predict that this would be the case. A question that our group was also very interested in answering was, it is well known that drugs that are used for the management of hypertension, and especially the management of hypertension and prevention of progression of kidney failure, so in kidney disease patients, ACE inhibitors and angiotensin receptor blockers are widely used. And we wanted to ask the question of whether the use of these drugs actually in any way affects the expression levels of ACE2 in the kidney. This is, again, a question that had been asked a lot in discussions around that time in the early days of the pandemic, and we were hoping to be able to find an answer. So we looked at the data set that we had. We had a limited amount of metadata on these patients, but from the small cohort of patients who we looked at, we could see that there were some trends toward higher expression of ACE2 in patients who were receiving these drugs, ACE inhibitors and angiotensin receptor blockers. Although I think it is important to say that this study is not well powered enough to draw firm conclusions about this. And I think what I learned from this is that we need to add metadata to our HCA and layer it in a way that we can begin to answer more and more of these questions when these questions arise. And so I think it was an interesting exercise that has led us to this important realization, this important lesson. This is from an interesting and well-written perspective on the kidney and SARS-CoV-2 by a member of our community, Ben Humphreys, and his colleague. And it really speaks to the fact that, again, the secret life of SARS-CoV-2 in the kidney is meant to say, well, we do have expression of the ACE2, and we have detected, as shown in that New England Journal paper I mentioned, active virus particles in the kidney. So it's possible for the virus to directly have an effect. But it is equally possible that systemic inflammation, endothelial damage, hemodynamic instability, all of these things in the hospitalized acutely ill patients can, of course, affect the tubular epithelial cells in the kidney and cause acute kidney injury. And so we are probably dealing with a multifactorial process. And so I think both are true. There's both a direct effect of the virus as well as kind of a bystander effect, if you will, in some cases. So I wanted to switch gears for a moment and give you a surprising twist to the story of thinking about the kidney and its connection to SARS-CoV-2 that in some unexpected ways led us back to the lung. And that has to do with MUC1, a molecule that my lab has spent some time focusing on, and how that led us to an insight that has some therapeutic implications. And so for several years now, my lab has been studying mucin-1, a glycoprotein present on most epithelial cells in the body. And I'm showing you here a picture of wild-type MUC1 in kidney epithelial cells. The reason we were doing this is because we were interested in mutations in MUC1 which cause an autosomal dominant kidney disease, and we were interested in studying the mechanisms that underlie that disease. And of course, we knew very well that MUC1, the wild-type version of MUC1, is highly abundant in lung, in fact, more so than in kidney. And this is something that was kind of known to our team, setting the stage for the insights that will come in the next couple of slides that I will share with you. One of the insights came from our colleagues in China. So there were several discussions that I was involved in earlier on, colleagues were involved in, where our Chinese colleagues who were taking care of patients in the ICU were mentioning how incredibly difficult it was to ventilate these patients. And part of the problem was that the mucus was exceptionally thick in these patients, suggesting that there was an upregulation in the mucins as part of this disease. And in fact, this is from a study in the Journal of Medical Virology from out of China that describes bronchoscopies in ICU hospitalized patients and shows an upregulation both at the mRNA and the protein level from mucin-1 in these patients. And so this was one piece of the puzzle that triggered us to think about MUC1 in the context of COVID-19. The other came from all of our communities' joint efforts on building an atlas for COVID-19. And this is work that was spearheaded very ably by Christoph Moos. Also significant contributions from Aishwarya Subramanian, all supervised by Aviv Regev, of course, in the early days of the pandemic. And one of the things that was identified in looking at cells that are double positive, as we call them, that are expressing ACE2 and TMPRSS2, responsible for the entry of virus into cells. At least as far as lung epithelium cells go, MUC1 was part of that gene module that predicted the double positive cells, as shown here. And so again, that gave us another piece of evidence that perhaps MUC1 was actually involved in the pathogenesis of COVID-19 in the lung. And so armed with that understanding, my group also realized that we might be the only group in the world that has actually done an entire screen of the repurposing drug set that is available, about 4,000 compounds, many of them FDA-approved drugs. We had done a screen looking to find removers of the mutant version of the protein, shown here in green, which is another story. It's intracellularly trapped and does not participate in producing mucus at the apical surface of epithelial cells. But in doing so, our control was actually to look at the wild-type version of the protein. We were looking for drugs that do not remove the wild-type version of the protein. In doing so, though, we potentially could have identified drugs that do remove the wild-type version of the protein efficiently. And we wondered whether, if we went back into that data set that we had generated, whether we would be able to identify an FDA-approved drug that removes MUC1 from the apical surface of epithelial cells. And so we did exactly that. I'm pleased to say that this work has just been accepted and will be coming out soon, so it has been peer-reviewed, but it was looking back at our data set of 3,000, almost 4,000 compounds. We looked for reducers of MUC1 from the surface of epithelial cells. We looked for compounds that are not toxic. We did several profiling counter screens looking for things that do not affect the transcription of the MUC1, but rather the protein levels. And we identified 4 compounds that were fulfilling all of these criteria and all of these counter screens, finally reaching the singular FDA-approved compound in this list, which is a drug called R406, which is the active the active compound of a drug known as fostamatinib, that was FDA approved in 2018 for idiopathic thrombocytopenic purpura, a completely different autoimmune disease, and nevertheless seemed to be very efficient at removing MUC1 from the surface of epithelial cells in a dose-responsive fashion. We wanted to understand this further, and so we did some in vivo work looking at the lungs of mice with acute lung injury. This is not not SARS-CoV-2 infected mice, but this is mice in which ischemia reperfusion injury induces an acute lung injury. MUC1 again is shown to be upregulated on the surface of the epithelium in the alveoli of these mice. And of course, treatment with fostamatinib showed a significant reduction. Again, suggesting now in vivo that there may be some merit to the idea of using fostamatinib for the treatment of acute lung injury in these mice. And so this is just a summary.
And I'm sorry, unfortunately, I'm sorry to have to stop you with such an interesting story.
It's just the end of the story. Yeah, absolutely. The only thing that I would like to add is that while I have no financial relationship, there is a clinical trial that is now starting based on these findings. So thank you so much. Thank you.